see some of you were there. So I will talk about more or less the same thing. So I will explain how it is different and how it is similar. So the angiogenesis is the formation of blood vessels, but in fact there are two different mechanisms to explain the formation of blood vessels. And these two mechanisms are this is the pointer. Somewhere here. Okay, vasculogenesis. So it's essentially from the differentiation of angioblasts. So we have some cells interacting with the matrix, and they spontaneously organize to form a network. So this is the vasculogenesis process. And the angiogenesis is quite different, because in this case, we already have an existing vasculature, and it is sprouting, and some new vessels are growing, and they connect to each other, and they create a bigger network. So this is occurring at a much later stage, so it's already a mature vascular network. And so the two mechanisms are related, but there are some strong differences in between them. So the talk we had 2 weeks ago about what described Fred, in fact, is something in between the two processes. It was somewhere in between vasculogenesis and angiogenesis, because he started by some non-existing vasculature, and he explained how this non-existing vasculature could sprout as in angiogenesis. So it was somewhere here. So vasculogenesis has been modeled for a long time now, and mostly the approach to model vasculogenesis were mechanical approaches, because it's basically interactions between the cell and the fibers, and with traction forces to organize the network. And in the other hand, angiogenesis was more led by the migration of cells towards some source of growth factors. So the approach was much more molecular. So there were two different ways of approaching the two mechanisms. One was mostly mechanical, and the other one was molecular. But in fact, there's no reason why to separate the two, because the mechanical effects exist in angiogenesis, and the same for vasculogenesis. So what we try to do is to investigate in angiogenesis the mechanical effects and how they affect the structure of the vasculature. So first I will present some experimental results that we did on vasculogenesis, so also called in vitro angiogenesis. So what we do is we have a matrix of fibers. It was a fibrin, the fibers were fibrin, and we put some cell on this matrix, some endothelial cells, and we see with time that the cells spontaneously organize to form a network. So what we see is that the cells, they don't really migrate, but they pull on the matrix, and they deform the matrix, and then they are convected, and they rearrange the matrix. And we see here what we call some lacuna, some holes. And Fred described these holes as in a 3-dimension, and in our case, it's just a 2-dimension process, because the matrix is so thin that nothing can happen in the 3rd dimension. So it's basically organization in the 2 dimensions. So we see these holes appearing, and with time, they expand, and the cells are all along the cords, and they form what we call the vascular cords. So at a later stage, they form some ridge in between the lacuna, and then they twist, and they form some tubes, and which are the vascular cords. Okay. So there are 4 main stages in the process: the appearance of lacuna, the growth, the fusion, and then the stabilization of the network. So we have been studying, in fact, the influence of the biogel rigidity. So we changed the rigidity by increasing the amount of fibrin in the gel. So with the low concentration of fibrinogen, then we have a smooth network. And if we increase the concentration, then we have what we call rigid networks. So what we have been looking at here, it is the shape index, which is an indicator of the tension force inside the gel. So here is the lacuna, and we measure the long and short axis of the lacuna, and it gives us the index. And the more the index is high, the more it is deformed. So 1 is the perfectly circular lacuna. And what we see is that for smooth gel, so there are 4 bars in each plot, which means it's 4 different times. So with time, we see here that the deformation is increasing for soft gels. And then for intermediate gels, we also have the deformation, but it's much higher. It's a much bigger deformations. And for rigid gels, we also have large deformation, but here, at this point, we see a sudden decrease of the shape, which means it goes back to a circular shape, and it is due to the traction force, which are so strong that the lacuna break and they form a bigger one, which is more circular, and then other tension form, and so on. So is it clear? Or you don't hesitate to stop me. So, okay. So it's what I explained. So the deformation is much bigger for intermediate biogel rigidity. So there is an optimum for rigidity to have this structure forming. And here the decrease is due to the fusion of the lacuna to increase the tension.
Yeah. Did you test the statistical significance of that downward thing?
Sorry?
Did you check the statistical significance of that downward time point? That effect on the stiff matrices of it going down?
I'm not sure I understand your question.
A statistical test.
Yeah.
That's real. That's not just.
Not an artifact. Yes, yeah, it has been measured on plenty different. So it's we are pulling experiments here. So what you see on the axis is the variation that we observe on the experiments. So it's a robust process. It's occurring. Yeah. So we've been able to derive this graph. So sorry, it's a bit in French. So the influence of the biogel rigidity. So when it's flexible, then we have some weak tension, and the kinetics of formation of the cords is very slow, and we have sometimes no network formed at all. And if we increase the rigidity, then we have some equilibrium tension, and it's a faster kinetics with very stable networks. They can last for several days. And when it's rigid, there are strong tensions, and as we have seen, we break the cords. And so the kinetic is slowed, and they are very unstable network because they break or they stop to form. So what we do here, it's when we do these experiments, we don't know if it's the number of fibers which matters more, the rigidity of the amount of aggregation, not adhesion points on the fibers. So to remove one of these parameters, which means the number of adhesion points, then we just tilt the Petri dish to have a variation of the apparent rigidity. So it means that when it's thick, it's smoother than when it's very thin, which is close to glass. So here it's glass, in fact, and here it's very thick. So it means it's more flexible here, and the rigidity is increasing in that direction. And what we see is that we have essentially in the middle the cords occurring, and none are formed here. And here they don't develop. So still this optimum. So this has been modeled a long time ago by some PhD students in our department. So the model was initially proposed a long time ago by Jim Murray, and it was adapted to fit our experiments. So the basis of this model is to make the fibers and cells interact together. So we see that the fibers F and the cell N. So the fibers, they are just convected by the deformation of the gel. So U is the deformations, the displacement of the gel. And the endothelial cells, there are three mechanisms to make them move. So there's the diffusion, and this diffusion is biased by some strains in the matrix, in the gel. And we have some convections and aptotaxis, which means that they can move, migrate with making links on the fibers. And we have the mechanical equilibrium. So this mechanical equilibrium describes the continuum between cells and matrix pulled together. So this continuum has some property. So we have some traction in it, mainly due by the cell, essentially due by the cell. We have some linear elasticity and long-range elasticity too. So this describes specifically the extracellular matrix, and this term describes the adhesion of the gel on the Petri dish. So we have these models, and if we run the simulations, so we start with a homogeneous distribution of cells on the medium, and slowly we see some lacuna appearing and a higher concentration of cells on where the cords are supposed to form. So this is the corresponding experiments, and it fits quite well. So many parameters have been analyzed, so I won't go into the detail of this. But
what are your boundary conditions?
No idea. I didn't do the, the simulation in that
Because
case.
it looks very strange that on the boundary of elongated,
Yes, yes. Yeah.
which might not be cells.
No. No, it's a big hole here. The density is very weak, but red is high density of cell. So I cannot tell more about this because, yeah, I just took this from the thesis.
It should be a no flux, no velocity boundary condition.
Okay. So what I did, it's angiogenesis. I've never really worked in vasculogenesis except for in vitro angiogenesis experiments. So I've mainly worked on angiogenesis and especially tumor angiogenesis. So the mechanism is the following: a tumor starts to grow without any vasculatures, and when it's big enough, then the resources around are exhausted, like oxygen and nutrients, and so the cell becomes in a state of stress, and when they are in this state of hypoxia, essentially, they try to they release some growth factors in the environment, and the aim of this release is to attract to them the vasculature so that they can gain some new resources to keep growing. So we have these small bits representing the growth factors. They diffuse into the environment. They are detected by the cell forming the vessel walls of the vessel. Yeah, the cell of the vessel walls. So when the cell detects this growth factor, it triggers a mechanism, so they detach and they start growing and proliferating, and they move towards the tumor and form this. This is the angiogenic process. They form some new vessels that will feed the tumor, and the tumor can keep growing as much as it can. And so, okay, so that's it. And at this stage, it's much more dangerous because then the tumor cell can escape the main tumor to invade the organism using the vessel, the motorway provided by the vessels. So the model is essentially the same. The hypotheses are the same. So we still have the fibers and the cell interacting together. So in that case, the fibers, they are just degraded by some proteases that are produced by the cells as they migrate, the endothelial cells. And the cells, so they are the endothelial cells of the existing vasculature. They move through the same kind of mechanism. So there's passive diffusion in that case. So no mechanics was taken into account initially in those models. We have some chemotaxis, which reflects the fact that the cells are going to migrate towards the growth factors, and still the apoptosis component. And it's mainly driven by molecular factors this time. So we have the growth factors that are produced by the hypoxic cell of the tumor. So they diffuse these growth factors, and they are consumed by the endothelial cell while they migrate. And we have the enzymes. So this is this enzyme, which is used to degrade the fibers. And the enzyme is produced by the migrating endothelial cells and diffused in the environment. It's produced by the cell, and it's degraded. There's a decay rate, spontaneous one. So I will explain how this continuous model, it's a continuous model, can give some discrete simulations. So I will explain that in a few moments. So the main thing is that when we put this model we run this model, we just have to create a vascular endothelial growth factor, which is the attraction for the endothelial cells. So we initiate some sprout on an initial vessels, and we create this gradient. So we imagine that the tumor is somewhere here and produce this gradient of BUGF, and so it attracts the cells towards that directions. So it's mainly the gradient of growth factors that will drive the migration of the cell. And we introduce some probabilities, which are the probabilities for the network to branch. So this probability increases with the level of VEGF. So the higher it is, the higher the probability to divide and create a branch is big. So that's why with time, so this is different times, we have this network growing slowly and making some branches as we approach the tumor, and it's very dense at the end. So it's typically what we observe in a real case. So this is a picture taken from a dorsal skin fold chamber on a mice, and we see here the big tumor mass, and we see the vessels and the angiogenic network. So it looks quite similar. So as you've seen in the initial network, I go back, no, go back, sorry, wrong direction. So at the end of the simulation, we have this homogeneous network with all the vessels with the same diameter. But we can perfuse the main vessels, and then if we take into account the rheological constraints due to the blood flow, then we can create some increase or decrease contraction of the vessels to explain how we have some differences in the diameter of the vessels. So, but this is another model that I will not describe here. So what I would like to do, in fact, is to take into account the mechanical aspects because, sorry, I go back again. In this network here in 2D, there's no problem for two vessels to meet because it's 2D, so at some point, if they turn to, they can join. But if you run the simulation, that something that we did in 3D, then the probability to meet is very low. And so it's a real problem. So something was missing in the model. And essentially, we think it's due to the fact we don't take into account the mechanics, and the mechanics will make the tension will create some lines that the cell will follow. So that's what we want to take into account to explain what we call the anastomosis process, which is the meeting of the two vessels. So there are different stages this sprouting angiogenesis. So the first is led by the molecular cues, essentially. So it is the sprout initiations, which is due to VEGF, the sprout elongations, still due to VEGF, and then the elongations in response to cues, the external cues. So we start to enter the mechanical process. And we have the lumen formations and the anastomosis. So essentially this phenomenon that we want to describe, I think it's mechanical cues are key to describe this spontaneous process. So we try to integrate all these mechanisms. So to illustrate how it works, if we have some cells put on some, so it's a very famous picture, you have seen it already, I'm sure. Fred, maybe, yeah, you have. So cells are here on a silicon, rubber, whatever it was, very compliant matrix. And when we put the cell on this, we see them creating some traction force, and we see the wrinkles appearing on this matrix, and they are the lines in between the cells. So it's deformations of the matrix. And the cells, they just follow these deformations to a line and create some bridge in between them. So, yep, questions. So what we take into account in the mechanical aspect is also the fact that the matrix is degraded. So it means that the rigidity of the matrix is not constant since the fibers are disappearing. So we have some, the Young modulus that we use to describe the rigidity is just a function of the amount of fiber in the matrix. So we take this into account. And we also take into account the traction force on the matrix fibers. So the cell adhere to the fibers and they pull, and so they deform these fibers, and the fibers are aligned, and this will help the cells to move. So the cell will migrate according to this time the matrix deformations, as in the vasculogenesis model, the initial one. The gradient of growth factors, of course, it will still follow these gradients. And we take also into account the fiber density, because it needs the fibers to create some adhesion point to migrate. So the new equations is the same, but this time we introduce this strain tensor for the diffusion. So now I explain how this equations is a continuous one. I change it into a discrete one. So you may be familiar with this model. So it's called the hybrid discrete continuous model. It was initially developed by Sandy Anderson and Mark Chaplin quite some time ago. And so the idea is to just decompose these equations with the different factors and the different coefficients. So we discretize using the centered scheme for each parameters, variables. And we have this, so it's at this point it's quite simple. And then we can reorganize the equations so that we have we can assign some probabilities to move in each particular directions of the grid. So in the initial case, we had only 4 directions considered, but we introduced the diagonals to improve, to have more realistic networks. So our cells can be located somewhere on this grid, and so the traction force are either horizontal or vertical. So I will explain that. So we still rearrange, so maybe it's not really interesting here, but what we get at the end is that the probabilities, they have two components. So initially only the molecular cues, but now we have the diffusion, the strain tensor, which is appearing and which creates some mechanical cues that will make the cell migrate in one particular direction. So it means that when this probability accounts for the fact that the cell will migrate in the most appropriate directions, so where there is more growth factors or where the deformation will be the highest, so it just follows the best, the optimum route to migrate. Yes?
So you have the PDE. Where do the probabilities come into this at all? You have a deterministic equation.
Yes, but now we make it random. So we create these probabilities, we throw dice, and the probabilities, often we have a higher probability to move in one particular direction if the environment is more favorable or not. And the idea is just because we were not able with the continuous equation to describe the branching and the individual cell. And we wanted to look at this aspect in particular.
And can you write down a continuous model?
Yeah, it's existing. We can still solve the continuous one. It works perfectly well. But when we introduce then it will be better when I show the automaton that rules. Yeah, so here we are. This is the automaton. So what we do is we take a grid and we have the existing vessels. We plot them from biological images, for example, and we have some sprouts. And then what we do is we have the sprouts here. So the sprouts are the migrating cells, and for each of these cells, we apply the cellular automaton we've just seen before, so the equations with the probabilities, and each cell will move in the environment according to the best direction. So what we do is, as I explained, we take into account cell traction on the matrix. So it's very complex because plenty of adhesions are formed everywhere, so the shape of the cell is not necessarily circular. But we assume that we can represent it with an ellipse, and so it simplifies the problems because we just rearrange the forces so that they are under the shape of a dipole, and so it's easier to integrate. But more complex set can be done. So the full model is the following. We still have the cells and the fibers interacting together with this diffusion biased by the strains. We have the growth factors and the matrix enzyme, the same. And we had this mechanical equilibrium. But to make it simple first, we just consider linear elasticity in the matrix, exactly as Fred did. So we are at the same point at the moment. And we have this adhesion still on the dish and the tractions of the cells, so that now we separate comparatively. This is purely the matrix, and the cells are externalized in the system. It's not a continuum anymore. So we discretize the first equations, so to rule the migration of the different cells. And we see that we have a discrete cell, so n is either 0, 1, there's a cell or there's no cell. And so this cell will appear here to consume the growth factors and will appear here to produce the enzyme to degrade the matrix. And it will appear here because it's creating the traction force on the matrix. So this is the linear elasticity, which is given here. And we see that the Young modulus is depending on the amount of fiber in the matrix. And here, E just represents the orientation of the dipole. So this slide just to show the shape of the deformation tensor. So it's very classical. And we use something, we are not very happy with that, but it's the first tensor that was proposed by Daphne Manusaki at the time and Jim Murray. So we just use it, but it's okay for now, but we will improve that. So it just describes the preferred movement of the cell along the regions where the matrix fibers are compressed and aligned, and it is proportional in each direction to the degree of fiber alignment under the effect of the deformation due to the traction. So we compute this in the following way. We first compute the cell positions on the cellular automaton. Then we compute the concentration of all the quantities, proteins. Then we calculate all the mechanics. So we have the Young modulus, which depends on F, So this is the input. And then we compute the traction force. So it deforms the matrix, and then it creates some strain, and then we can have some probabilities, which are related both to the molecular cues and to the mechanical ones. And then we go back to update the cell positions, and we do that, and we run the simulation. So these are the simulation results. So first, we just study the general influence of the mechanical inputs. So what we did is we did the simulation. So here is the parent vessels with some initial sprouts, and the network is growing in that direction. And this, so what we see is the angiogenic network formed. And here it's the fiber density, which is also the map of rigidity. So we see when it's red, it's rigid, and when it's bluish, then it becomes degraded. So it's more flexible. And so what we see, if we involve the mechanics and we don't involve the mechanics, we see some difference. And essentially, the density is higher without the mechanics. And we have a reduced matrix degradation with the mechanics. So it means here that mechanics is a ordering force, which means that it pushes the cell to meet and to organize, whether here it's much more random. And so that's why cells are moving more randomly, and so they have time to degrade the matrix, and so there are some difference appearing. And it goes in the right direction, because what we want to explain is that it's easier for the cell to meet. So we did exactly the same experiment again, but this time to remove the other process. We remove the chemoattractions. So we just allow the cell to move randomly on the matrix here. So maybe I might have a simulation to show you. So it's just we put some 4 cells initially, and they are left in the middle, and they just grow as they want. And what we see is that they degrade the matrix, and they just move randomly, and they have time to degrade and all that. Okay, so it's very fast. Okay, I can stop. Now it's the idea. So what we see, it's even more obvious here in this simulation, because here it's very well organized with trajectories mainly in long straight directions, and here it's very messy. So it is explained, in fact, by the fact that traction force allows the cells to connect, to find their way in this network, and to connect and create the bridge that we need in the anastomosis process. So we did this simulation again, this time we modulate the traction force of the cell on the matrix. So what we see here is a superimposition of 100 simulations, and in red is where the trajectory has been reinforced, where there is lots of trajectories going at the same point, and grayish is the trajectories that are not, is it clear or no? You superimpose the trajectory, so when it's very bright, there are lots of trajectories, and when it's not, there are very few. And so we did that because, as I said, it's a cellular automaton, so we cannot perform any mathematical analysis. So the only way to do that is to make a lot of simulations and then to make some statistical analysis on the simulations. So we start with the weak traction force, and we see there's no big difference with no traction force at all. And when we increase the traction force, is 20, then we have less degradation, a more organized network. So we go on increasing, so it's too big because now we don't have a functional vascular network, and at the end there's no network at all because the cell is pulled too strongly and nothing's happened. So we did it again without the chemoattractions, so it's exactly the same superimposition of the network. So here it's messy, and when we increase, it becomes better, and until it doesn't work anymore. So then we still played with this system. So now we've been studying the influence of the matrix rigidity. So what we did is we performed some simulation on this domain. So where it is white, it's very smooth, and where it is red, it's very stiff. And what we see, can run it, is that the cell, they can cross very easily from white to red, but not very easily from red to white. And this is a well-known phenomenon, which is due to gyrotaxis. So it means the cells, they prefer to go on stiffer substrates. It's easier for them to move. It's like moving in the sand. Maybe, yeah, Jocelyn did this image once, no? That when you move in the sand, it's difficult. And so you can't go, but when you find some yeah, better, you call it soil, you can come across, and then, so it explains how you can reach this. Okay. So it's easier. And then we've been playing with some gradients. So what we did is we created some gradient of rigidity here. So it's a very strong gradient here and a low gradient here. And this place is smooth, very smooth. This is slightly higher rigidity and the two gradients. And we just put a cell right here in the middle and we let it go and see what happens. Should I put it here? So when it managed to find a gradient, then it goes straight into it follows the gradient. It's very easy. So when it's a high gradient, strong gradient, it's even quicker. It goes straight in straight line, no problem. And when it can find the gradient, then it's a random walk. So it moves in any directions. Okay. So that gives you an idea, random. Okay. And so I show you the same. I superimpose all the trajectories we got with this system. And what we see is that they mainly go into catch the gradients, they go in straight lines. And if it's very difficult for them to go in smoother parts, but very easy to go in more rigid parts. Okay, so the last thing we've been investigating is the influence of the matrix deformations, so the strains. So what we did is that we put some cells on the matrix. So they create what we see here is the deformation field created by the cell at the neighborhood of the cells. And we just put a cell in the center. All these cells are static, they don't move, they just create some strains on the matrix. And we just release the cell in the center and let it go. And we superimpose all the trajectories, and this is what we get, this cloud of trajectories. And what we see is that the trajectories are mainly trapped in between the set of deformations. So the strain acts as a boundary for the other migrating cells. So it's also a way to find their way. So the strain in both directions. So we tried to make a little bit of quantification of all this work. So what we did is to plot the mean cell velocity as a function of the cell force intensity for different Young modulus. And what we see is that the traction force influences the cell migration velocity. The higher it is, the more the cell is able to move quickly. But the impact of the Young modulus is very small. So when we put it in the degradation range that we feel was realistic, from here, from the degradations of 30%, it corresponds to a degradation of about 30% in the experiments. And there's only a very limited effect. The velocity is nearly the same in each case. So we decided to increase the Young modulus range, a much higher one. And so we see that there is, in fact, an impact on the velocity, but which is located somewhere here. So we have a sharp decrease. And the problem is that, oh, no, I explain later. So, okay, the impact of the cell velocity is observed for the much larger range. So then we've been studying the maximum strains as a function of the Young's modulus. And what we see is the strain is higher when the strain is higher with the cell traction force. So it's obvious when we pull very strongly, then we have big deformations. And the maximum strain generated by the cell on the matrix is reduced as the rigidity increases. So the matrix is less compliant, so it becomes more and more difficult to deform this matrix. So yeah, we did this, but we are quite not completely finished with this study because we have only worked on arbitrary units because we used some previous models and we used the same parameters of the previous models and they were not describing exactly the same set of data. So we were not very looking at this carefully. So we have worked essentially in arbitrary units. So now the next step is really we need to quantify with the experimental data that we have now on angiogenesis to have real values of Young modulus, realistic ones, to be able to replot these curves and define if we are right or wrong. Okay, so there are plenty of things that we still need to do. We need to find first a better relationship between the fiber density and the Young modulus, because here we just took a linear relationship, which is clearly not the case. We need to integrate some viscoelastic effects. So on this, probably I will work with Fred, because he's looking into it also. So we need to modulate the traction force with the matrix rigidity, and for that we have plenty of experimental data. It's a very well-known phenomena, so it will be, I think, easy to find all that we need in the literature. And we need to test a more adapted diffusion tensor. As I explained, we are still using the one proposed by Daphne many years ago, but it's not adapted to our new configurations. And we also need to consider more explicitly the fibers in the matrix, because here we do not describe the fiber itself, we just describe a main orientation. So we would like to do that, to integrate the fibers and see how they are deformed in the system. So this is something that a colleague of mine, Arnaud Chauvière, in the department is interested in. Okay, so I want just to finish to thank my collaborators. So Arnaud Chauvière, who has been involved in this project, and Simon Lefloc, who is working in the mechanical departments, and we did most of the, who was the advisor on the mechanics, because I'm not really a specialist in mechanics. So, okay, thank you for your attention.
Questions? You have also vascular formation in fluids, right? So do you think that your model could be adapted to take into account some hydrodynamical effect rather than elastic effect?
Yeah, I think so. Yeah.
Because in fluids there cannot be the elastic effect, and so something else has to be happening in order.
Yeah, we could trick it to take into account any external force to induce the migration. So we need to integrate, I don't know, maybe some fluid force equations. But yeah, but I don't see in which experiments are you?
Well, I was thinking of the work by this Italian group, Preziosi and collaborators, and they propose a similar model, but with no elastic effects, so just fluid.
Okay.
And they get vascular formation.
Okay, because in our model, maybe in this one. Here. Here, as I said, we take into account the blood flow into the vessels, but the flow is inside the vessels, and is directing the branching.
Yeah, I guess they take into account that, which apparently has an important effect.
Yes.
But also the exterior flow.
Yeah. No, I have never tested, but I think it's not. It's doable from this kind of model. But we did take into account the flow inside the vessels, and it's very important because the shear stress inside the vessel, they can induce help to branch. So they are playing a big role in branching.
So is the cell automaton coupled to fluids in the solvers?
In this case?
No. In general?
Yeah. In this case, it is. We couple the flow equations with the cellular automaton. So we compute the automaton. When we have a loop forming, then it's flowing. So then we stop the automaton. We have the fluid dynamics taking place, and then it goes on. When it's stationary, then we go on with the automaton. And no, the framework is very easy to use, in fact. So that's why I'm very confident that we can use it for other type of coupling.
How are you driving that fluid flow? Is there a pressure gradient somewhere?
Yes, you calculate the pressure at all the nodes of the systems, and then you have some pressure drops in each vessel. So you have your flow going, and you update at each time a new loop is formed. Then it's redirecting the flow. So you have to recompute. So the flow modeling takes a lot of time, computational time, because you have to wait for stationary states. each time.
Is there any chance to couple this with a tumor growth model?
Yes, we did it.
You did it?
Yes, yeah, I haven't presented it, but I will present this at the workshop in December.
Sure.
So the coupling with the tumor model.
I see.
Yes, yes. Yeah.
So then can you explain the bifurcation and when the tumor grows, right, it starts to deviate from the ball. When it's small, it's basically a ball, right?
Yes.
And then at some point there happens a bifurcation, right? So it gets other shapes.
Yeah.
Is that partly caused by
The vasculature.
yeah?
Yes. Yeah, it is. Yes. It depends where the vessels are. So it will drive the growth of the tumor. It will grow mainly towards the big the
yeah.
vessel.
Okay.
And
That
so
makes sense.
so the shapes change. So yeah. So I have simulation. So essentially, it's the same grid that we have, and we imagine that the tumor is somewhere. We put some tumor cell in each grid elements, and then here we can put a tumor and then see how it grows. And so that's what we did, and it works fine. Yeah. Yeah. So that will be in the next talk.
Any other questions? If not, we then can give it a
You have. Sorry. Okay.
Very good.